to each and everyone. I am Ivan Sarel Vigalanga from grade 11 STEM section Linnaeus. For today's video, I'm going to talk about genetic engineering and genetically modified, also called GMO, and steps on creating an organism. Okay, let's define what is GMO. A genetically modified organism or GMO is an animal, plant, or microbe whose DNA has been altered using genetic engineering techniques. My Scrofa bos taurus. Scrofa came from the word sus, Scrofa bos taurus, which means pig, and bos taurus, which means cow. This GMO, it was made from the DNA of a cow and a pig. An egg was taken from the ovary of a cow and a cell was taken from the pig. This GMO will benefit us, especially the farmers, since it can give the birth to multiple babies. Normal cows only can give birth one offspring and it takes along 9 months. I want to create this GMO since it will help us to avoid extinction and it can boost biodiversity. It might be able to eat a certain food that its parents species cannot, or maybe it can thrive in a different habitat, and it includes passing along favorable traits and prolonging the survival of threatened or endangered species. These GMOs, top part of the head was like that of a calf, but the bottom jaw, eyes, and the ears were those of a pig. There was no neck, the head being set directly on the body. The front leg has no shoulders, and the tail, like the one of a pig, was attached to one of the hind legs. To produce a GMO, new DNA is a transfer into organism cells. Usually, the cells are then grown in this culture, where they develop into organism, the next generation of organisms produced by this organism and will inherit the new DNA. The genome contains genes and regions of DNA that usually carry the instruction for making proteins. It is proteins that gives the organisms their characteristics. Identify a trait of interest. In order to identify desirable new trait of scientists, most often look to nature. Successful discovery of a new genetic trait or interest is often a combination of critical thinking and luck. For example, if researchers are searching for a trait that would allow of organism to survive in a specific environment, they would look for organisms that naturally are able to survive in the specific environment. Or if researchers are aiming to improve the nutritional content of an organism, they would screen a list of animals they hypothesize to produce a nutrient of isolate the genetic trait of interest. Comparative analysis is used to decode with a part of an organism genetic makeup contains the trait of interest. The genome of animals with the trait are compared to genomes in the same species without the trait, with the goal of identifying genes present only in the farmers. The genomes of different species with the same trait may be also be compared in order to identify gene if there is the base or genetic information. For comparison, scientists will purposely delete or knock out parts of the genome of interest until the desired trait is lost, thereby identifying the genes that lead to the trait. Insert the desired genetic trait into the new genome. Altering the genome of organism is difficult due to their rigid structure. Many biotech companies use gene guns that shoot metal particles coated with the DNA into animal tissue with a 22 caliber charge in biotechnology researchers. It's a common to genetically engineer bacteria to produce a desired protein. This is done by using enzymes to cut and paste a DNA strand of interest into a plasmid, which is a small circular molecule of DNA. Bacteria are then shocked using heat or electricity so that the cells accept the engineered plasmid by modifying to ferment sheet, which is easier to modify than plant seeds themselves. Researchers maybe use the bacterial natural or invasive behavior for inserting desirable traits into a crop of a genome. Growing the GMO. After a genetic trait has been successfully inserted into our organism genome, that modified organism must then be able to grow and replicate with its newly engineered genome. First, the genome type of the organism must be checked so that researchers are only propagating organism in which the genome was modified correctly. Recombinant DNA is engineered through the combination of a two or more DNA strands, combining DNA sequence and which would not normally occur together. In other words, selected DNA is inserted into existing organism genome such as a bacterial plasmid DNA or some other sort of vector. The entire genetic engineer process is basically the same for any organism.